Let's start with a simple but critical question. What if your drug product is perfectly developed, but something from the packaging silently contaminates it over time? That's exactly where extractables and leachables or E&L studies exist. In this presentation, we'll explore how USP guidelines help ensure patient safety by identifying and controlling these hidden chemical risks. Hi, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, Canada. As a CRO CDMO focused on complex drug molecules, Resolve Mass Laboratories delivers precise, science-driven and solutions for peptides, impurities, and regulatory critical analytical challenges. To understand E&L, let's break into two simple ideas. Extractables are compounds that could potentially migrate from packaging materials when exposed to harsh conditions like heat or strong solvents. Think of this as the worst case scenario. On the other side, leachables are compounds that actually migrate into the drug product during real storage conditions. So in short, extractables predict the risk and leachables confirm the reality. The critical importance of E&L. Now, why E&L is so important? First and foremost is the patient safety. Even trace level chemicals can cause toxicity or immune reactions. Second, drug efficacy. Leachables can interact with the formulation and affect stability. And third, regulatory compliance. Health authorities expect complete control over unknown compounds. So E&L isn't just testing, it's a safeguard for both patients and product quality. USP regulatory framework. USP provides a clear roadmap through two key chapters. USP 1663 that focuses on extractables, how to design and perform these studies. USP 1664 focuses on leachables, how to monitor them in real products. Together, they promote a scientific, risk-based approach, guiding decisions across the product lifecycle instead of applying a one-size-fits-all method. Extractables assessment. So in extractable studies, we simulate extreme conditions to understand what could come out of the materials. These includes using strong solvents like ethanol, hexane, combined with high temperatures. The idea is to generate a complete chemical fingerprint. To do this, we rely on advanced analytical tools. For example, GCMS for volatile compounds, LCMS for non-volatile compounds, and ICP-MS for elemental impurities. This data set becomes the foundation of everything that follows. Real-world leachable monitoring. Now we move from theory to reality. In leachable studies, we test the actual drug product over its shelf life under real storage conditions. Samples are analyzed at multiple time points, often detecting compounds at extremely low levels, even parts per billion. Here is the key advantage. We don't just start from scratch. We use extractables data as the targeted roadmap, making detection more efficient and meaningful. Now let's talk about safety thresholds. Once compounds are detected, the next step is evaluating their safety. This is where thresholds come into play. The analytical evaluation threshold, or AET, defines what needs to be reported and identified. The safety concern threshold, or SCT, defines what requires toxicological assessment. These two thresholds ensure that the efforts are focused only on compounds that could pose real risk to the patients. Now let's talk about implementing a risk-based approach. USP strongly emphasizes a risk-based strategy. Not every product requires the same level of testing. For example, injectables and inhalation products are high risk because they directly enter into the systemic circulation. On the other side, oral tablets or topical products typically carry lower risk. So instead of over-testing everything, we prioritize based on exposure, route of administration, and the material interaction. Common industry challenges. In real-world scenario, E&L testing comes with challenges. One major issue is unknown peaks, compounds that don't match any unknown data, any known database. Then there is material variability, where packaging components differ between batches. And finally, finding the right balance, avoiding excessive testing while still meeting the regulatory expectations. This balance is where scientific judgment becomes critical. Differ between batches. And finally, finding the right balance, avoiding excessive testing while still meeting the regulatory expectations. This balance is where scientific judgment becomes critical. So finally, we are at the conclusion. So to summarize, E&L follows a clear 4-step workflow. First, identify potential compounds through extractable studies, which is worst case scenario. Second, monitor actual migration through leachable testing. And third, assess safety using toxicological evaluation. And finally, control the risk to ensure patient safety. At its core, E&L is not just about compliance, it's about delivering safe, reliable medicines every single time. Partner with Resolve Mass Laboratories to overcome your toughest drug development challenges and move your program forward with scientifically robust solutions. Thank you so much for watching this session. Goodbye for now and see you in the next one.